DNA sequencing is the process where we read the exact sequence of 4 building blocks or bases that make up the DNA. There are some known methods of DNA sequencing. Sanger sequencing, sequencing by synthesis and concentration-based DNA sequencing. DNA is denatured by applying heat. DNA splits into a complementary strand and the template strand for sequencing. Primer is annealed to the template of DNA. DNA is spread equally in 4 reaction vessels and DNA polymerase is added into this. Only one type of ddNT piece is added to each reaction vessels. Polyacrylamide gel electrophoresis is used to sequence the DNA. The smaller, lighter fragments of DNA further migrate to the bottom of the electrophoresis plate. This gel is used for high resolving power and separate DNA by its lengths. Sequencing begins with producing the first read. Each nucleotide is included in template for growing chain. Millions of clusters are sequenced in a parallel process. The read product is washed away. Index 1 primer is introduced. This read is generated and washed off. The ends of the template is protected and binds with the second flow cell. Index 2 is read in the same way and washed off. Polymerase extends the second flow cell and forms a double-stranded DNA which is linearized and the forward strand is washed off. Read 2 begins with the read 2 primer. Read 2 product is washed off. Clusters are read. Reaction chamber is equipped with electronic heating and cooling system. Each DNA fragment is amplified by varying the temperature from 95 degrees centigrade to 40 to 60 degrees centigrade and finally to 72 degrees centigrade for a certain number of cycles. In the sequencing reaction, the DNA cluster is tested by adding one of the nucleotides at a time. If the nucleotide is complementary, a change of concentration could be traced. This change could be tested directly in any part inside or outside the sequencing chamber.